fight or flee yet. It's still just tyrosine. But if we add another OH group, then chop off this carboxyl, add a methyl, we get adrenaline. And that's the hormone that is like our fight or flight hormone, among other things. It's not really that simple. But anyway, so tyrosine, this is the amino acid tyrosine. And so we normally think of amino acids as protein letters, and tyrosine can serve as a protein letter, but it can also like moonlight as other things. Typically after some modification, it can act as a neurotransmitter, so something that nerve cells send to other cells, and as a hormone, so something that glands send. And these are ways that the byproducts of tyrosine can serve as these like chemical messengers to tell your body to do things like fight, flee, fright, rest, digest, all of that various stuff. And so all of those things can be helped, messages can come from tyrosine. And so basically what happens is, so tyrosine, like all amino acids, it has that like amino part and the carboxyl part that's going to let them link up to other letters, and then it has this unique part that's going to stick out, this R group, or this side chain. So yesterday we looked at phenylalanine, which was basically this. With tyrosine, we're going to add this OH group, this hydroxyl group. This is going to have some different consequences. We'll get more into it later, but one of the things is it's going to make it polar. So basically all of this is kind of blah. The side chain of phenylalanine is kind of blah. It's interesting, but it's all carbons and hydrogens. And carbons and hydrogens, they share electrons fairly. And so this is going to be very, like no partial charges, no full charges, no nothing that water likes, because water has like partial charges because of the oxygen pulling electrons from the hydrogens, and the hydrogens are partly positive, the oxygens are partly negative. So water likes to hang out with other partly or fully charged things. We call those things hydrophilic, and then we call things that water doesn't have any like interest in hanging out with hydrophobic, and so those things tend to get like clustered like inside of proteins and that sort of thing, which we call like the hydrophobic effect. So basically with phenylalanine, it's like super hydrophobic, so it would make like a crappy messenger, like because you can't get it through your body, right? It's just like water would not want to like dissolve it. So like with dissolving, you have to like form a full coat of water around it, and that would take a lot of water molecules, take up a lot of molecules out of their water networks in order to like form this like cage or clathrate around it, which wouldn't be good. So if you add the OH group, now you're a little more soluble, but still not really soluble enough to act as a good messenger throughout your body. So what's going to happen is to turn this into like a chemical messenger, what you're going to do is you're actually going to add another OH group. You're going to add another hydroxyl group, and this is going to make it L-DOPA. And so the L is coming from, because we have, as we talked about before, like with amino acids, we have stereochemistry because this carbon is attached to 4 different things. And so you can imagine this part being like sticking forward, or this part like swapping this part with hydrogen, so it'd be like sticking out. So we have like the L and the D forms, and we're only going to, our bodies use the L forms. And so since we're starting with L-tyrosine, if we make a change to that, we're still going to be in the L, so we're going to have L-DOPA, and we're going to have the OH group there. So this is going to make it more soluble, yay! And it's also going to make it so that we give it a new name. We give it, we call it L-DOPA, but we also call it a catechol. So a catechol is where you have 2 hydroxyl groups on a benzene ring. So this is the benzene ring, like we were talking about yesterday. It's aromatic, so basically you have shared like partial double bond character throughout here, and you have shared electrons around this ring. So all of these atoms are joined together by sharing pairs of electrons. So 1 pair for a single bond, 2 pairs for double bond. You have a little extra, you can donate to like this electron commune, through this like electron delocalization or resonance, and this happens in a ring. We call it an arene or an aromatic compound. And so with phenylalanine, we saw that we had this benzene ring. With tyrosine, now we add this OH. If we add a second OH, now we have 2 OHs on a benzene ring. We're not disrupting the aromaticity because we're basically just swapping out the hydrogen for the oxygen. And since this is still a single bond, it's not disrupting the ability to donate that extra electron to the rest of this aromatic ring. But when you add this OH group on, so now we call this a catechol. And since we have the amine group, we call it a catecholamine. So then to process it further, what happens is we're going to remove this carboxyl group. So we're going to decarboxylate it. So we still have the amine group, we'll still have the 2 OH groups, But at this point, we will, so it will still be a catecholamine, but now will be a catecholamine called noradrenaline or norepinephrine. So some places in your body, things will stop there. Most places, things will stop there, and it can act as like a sympathetic nervous system thing. So this is more of like the rest and digest stuff. But in your adrenal glands, so these little glands that sit on top of your kidneys, add on top of renal kidney, these glands can take things further. Like in the medulla of it, it can take things further and process it by adding a methyl group onto the ring. And so this is going to, when it adds that methyl group, now you're going to get adrenaline or epinephrine. So epi means on top of kidney, nephrin, kidneys. So basically adrenaline and epinephrine mean the same thing etymologically, but just like different sources and that sort of thing. So depending on where you live and that sort of thing, you might see different terms. But the basics is that now that is going to act as like your fight or flight hormone. And again, this is like super oversimplified. Things are way more complicated, and what's going to actually happen is depending on like a cell type and the various forms of receptors they have, like adrenergic receptors. So I talked more about the catecholamines in another post in terms of like their function and stuff. Today I'm going to go more into the synthesis, and so some of it is going to be like refreshed from that video I did on Halloween, and there's also going to be some new stuff, more specifically about tyrosine. So there's some other stuff that is cool about tyrosine as well. It can also be used to make like thyroid hormones. We make it from phenylalanine, so about half of the phenylalanine we talked about yesterday is actually used to make tyrosine, and it gets its name from cheese. Is day 8 of 20 days of amino acids, and we are at tyrosine. So tyrosine is coded for by UAC, and UAC every time that the ribosome, when it's traveling along the messenger RNA and it sees that one of those codon words, it's going to put in a tyrosine. So a tRNA holding a tyrosine is going to come and it'll add it to the chain. It adds to the chain. Phenylalanine and also like tryptophan, it is an aromatic amino acid. So basically there's partial double bonded characters around this ring. So we draw it typically as these alternating single and double bonds, but we can then draw like resonance structures, or sometimes we draw like a circle in between. Basically, these are all like the electrons are being shared fairly throughout, but we can't represent like the reality. So it's really like a hybrid in between these. And you have this like electron delocalization above and below the planes. So if we look at how we can classify amino acids, so these are all three of these were aromatic, but tyrosine's special in that it's polar. And so it's polar because it has this OH. And so it's polar because oxygen's an electron polar. So polar, that's just a way you could remember it. But basically, so these atoms, they're linking up through sharing pairs of electrons. And if they don't share fairly, then you can get a partial charge because the atoms, so like the individual carbons, hydrogens, oxygens, they have protons which are positively charged and stuck in place. And then they have these electron clouds, and electrons are negatively charged and they're like whizzing all around, and they're shiftable. So when they share pairs of electrons to form bonds, what's going to happen is that if one of them is better at pulling the electrons, so we call these electronegative, then they're going to get more of the electron density, but the protons are stuck in place. So these positively charged protons are stuck in place, but the negatively charged electrons can move. So when they move away from the hydrogens are going to be partly positive, and the oxygen is going to be partly negative. And so here we're showing it in water, but the same thing is also going to happen when you have it in a hydroxyl group, which is what we have here. So this is also called an alcohol group. And so you have this OH, and the oxygen is going to be pulling electrons away from both this hydrogen, but more important, but also from the carbon. And this oxygen is going to be partly negative, and it'll be like we can get it to react and stuff. So tyrosine, like
serine that we saw before, and like threonine that we saw before. We haven't looked at serine yet, but serine as well. It can get post-translationally modified through phosphorylation. So post-translational means that it's added on like after the protein's already been put into the chain. So after that happens, it can get a phosphate group added onto it. So more on that in the threonine post, but basically after this OH is going to allow it, because it has this like nucleophilic, it can like nucleophilically attack. Basically, it can pull off a phosphate group from ATP or phosphoryl group to make phosphotyrosine. And this is done with the help of enzymes called kinases. And tyrosine kinases are very important in like intracellular signaling. So often there will be like a signal that will arrive on the outside of like these tyrosine receptor kinases. So they're like membrane bound proteins, and they go through the membrane, and then like outside of the cell, it'll bind to some thing like like insulin. So insulin receptors, a tyrosine kinase. And so it'll bind, and then like inside the cell, the kinase will like phosphorylate and activate other things, and you can have these like cascades. And so tyrosine kinases, tyrosine receptor kinases are really important for growth and development. And if things go wrong, then like cancer can occur, because you can have like if they become detached from like the signal that they're supposed to be responding to, they can kind of like go on the fritz. And so that's why there's sometimes drug targets for cancers. But normally this phosphorylation is happening in this controlled manner, and it helps your cells like communicate, and it helps your cells like respond to outside stimuli. So it's really important. And so this is in addition to tyrosine's role as serving as a precursor to making those hormones. And so we'll talk more about the catecholamine synthesis. So I talked most about the catecholamine synthesis, but there are also other things you can do with tyrosine. And so if you go this, this is the catecholamine pathway. So you go DOPA, dopamine, noradrenaline, adrenaline, but you can also go a different pathway and make thyroid hormones. And so these are important for thyroid functioning, and these come from tyrosine as well. You can also go a different pathway from L-DOPA. You can actually get L-DOPA a different way using a different enzyme called tyrosinase. And this can lead to give you L-DOPA, and then steps later to give you melanin. So disorders that you can get, like mutations in enzymes in this pathway, that can lead to albinism, because melanin is like the colored pigment that gives skin that's pigment. And so if you have a problem with this pathway, then you can have problems with pigmentation. There are also other forms of disorders, metabolic disorders, like genetic diseases that people have that involve a problem with the breakdown pathway of tyrosine, as well as we talked about, phenylalanine. You can have a disorder that prevents the conversion from phenylalanine to tyrosine, and this causes phenylketonuria, PKU, but tyrosine, you don't want it to build up, because it you need it to make all that other stuff. And if it builds up, it can like form these weird side products and stuff. So there are different diseases associated with various steps in this pathway. So if all goes well, you get breakdown products that can enter both the tricarboxylic acid cycle, so the Krebs cycle or the citric acid cycle, be used to make glucose, so we can call it glucogenic. And you also get products like acetyl-CoA, that can be used to make lipids or to form ketone bodies. And so we also can classify it as ketogenic. So it's one of the amino acids that's classified as both glucogenic and ketogenic. And more on the differentiation between those in my talk about leucine. In terms of divs and naming, so the name comes from tyros, which is Greek for cheese. It was discovered by the German chemist Justus von Liebig in 1846, and he found it from casein, which is a protein in milk and cheese, hence the name. A couple of years later, Warren de la Rue found it in an insect, and then Emil Erlenmeyer, who's that guy that we get the Erlenmeyer flasks, those like kind of shaped flasks. Yeah, so he and Lip synthesized it. And so more on that on my blog and in this article. If we look at our phenylalanine, we can see that its side chain, it just has like carbon and hydrogen, and carbon and hydrogen share pretty fairly. And so this is going to be very nonpolar and hydrophobic. If you add this OH group, so oxygen is really what we call electronegative. So it's going to hog electrons. So it's going to hog the electrons that it's sharing with the other atoms. And this is going to make, so now you have this oxygen be partly negative and like this hydrogen be partly positive. And now you have something that water is okay with. So it's still not super water soluble, but it has, it's better than phenylalanine. And to make it even better to travel through the bloodstream, to make it even more water soluble, we can add a second OH group. And so this is done by this tyrosine hydroxylase enzyme. So remember hydroxylase, we're adding this hydroxyl, so this OH group. And this is done with this tyrosine hydroxylase enzyme. And it's going to add this second OH group. And when you have a benzene ring with these two OH groups, we call it a catechol. So that's where that cool name comes from. It's because it has this benzene with these two OH groups. And at this point, when we call these catecholamines because remember we have this amine group from our amino acid. So if we go to our functional group, we have an amine group, and we have this catechol group, which isn't on this chart, but we can see that it's a benzene ring and it has these hydroxyl, this OH group, two OH groups. So that's why we call these catecholamines. And you can see that although we're going to make changes, we're not going to change like this bottom part. So we're not going to take this away from being a catecholamine, and we're not going to change. We're not going to take away this nitrogen. So we can call all of these catecholamines. And so we're done this. So now we've encountered two of these two different enzymes already. Although if you start with tyrosine, then you don't need this one. But these both are using tetrahydrobiopterin as a cofactor. So a cofactor is basically something that an enzyme uses as a helper. So an enzyme is a reaction speeder upper. It can do things like hold the reacting molecules in the optimal position to react and stabilize uncomfortable transition states, provide give and take electrons, hold on to things, all sorts of different things. And so sometimes in order to do their work, they use, they bind to other molecules to help them out. And we call these other molecules cofactors. And I really need to organize my figures ahead of time better. So we call those cofactors. And so we can refer to, if an enzyme has a cofactor, we can call it, when it's alone, we call it an apoenzyme, enzyme by itself. But then when it has a cofactor, now it's a holoenzyme, so it's all whole. And so sometimes these are metals, sometimes these are organic, which are often vitamin-based, as we'll see in a couple in this case. And so these are cofactors can provide additional opportunities, so additional functional groups and different metal groups to help bind things better, all sorts of different things. So enzymes often use cofactors is the basic point I wanted to make. And so in this case, they're using this cofactor called tetrahydrobiopterin. Okay, so we've gone from our tyrosine now to L-DOPA. Okay, so L-DOPA, so the L, so this goes back to our discussion of chirality, so like handedness. So like the right hand versus the left hand, non-superimposable mirror images. And so almost all of the amino acids you'll encounter in biochemistry, except for like some cases with like some bacterial wall stuff, are in the L form. And so that's where this L comes from. So you have it attached in a way. You have this group, so you can see that this carbon, this alpha carbon, that's attached to 4 different things. If you have a carbon attached to 4 different things, then you have a chiral center. And so you can think of this like as being stuck, so it's sticking out at you, or so it's sticking back. And one version would be L, one version would be D, and all of the amino acids that we use are L. And so that's where the L part comes from. Okay, so now we've made a catecholamine. We have this L-DOPA, but we now want to make it like a neurotransmitter or a hormone. And so we've made it more soluble, but now we need to do more stuff to it. So the next step is done by another enzyme, DOPA decarboxylase. And so carboxyl, so remember, carboxyl group is going to be a C double bond to an O attached to an oxygen. And then this could be protonated or deprotonated. So we call those different forms, carboxylic acid or carboxylate, depending on whether they're protonated or not, but it's the same molecule that can be just, yeah. And so if we have a decarboxylase, it's going to remove a carboxyl group. So it's going to remove this carboxyl group. So we're going to remove the carboxyl group of the amino acid. So now it's no longer going to be an amino acid, but it still has that amino group, so it can still be a catecholamine. And so this decarboxylase is going to use a cofactor too. And it's going to use a cofactor called pyridoxal phosphate. You'll see this PLP a lot of places in biochemistry. And so if you've been following me a while, you might remember in the talk about the amino acid lysine. So lysine is another amino acid we've talked about. And so it's this, and it can form what we call shift bases. So it has this like amino group at its end, and it can form these shift bases. So basically it like double bonds to a carbon. And this allows it to kind of like hold on to this cofactor PLP, which is the cofactor we're talking about here. And so in this case, we were talking about lysine being used in transamination. So like moving an amine group from one place to another, which is involved in helping make amino acids from like sugar by breakdown products and stuff. But basically this shift base allows it to hold on to this PLP, and PLP doesn't just have to do this transamination. It can do all sorts of other things. And so one of the other things that it does is helps out with this decarboxylation reaction. So yeah, I just got this from this. There was this review article about all the different things that PLP could do. So you can see that it's forming this shift base, so it's holding on to that PLP like we saw before, but then it's going to switch that out so the enzyme is going to kind of hold that PLP in place. And then once the substrate comes in, so the L-DOPA, now it's going to hold on to that and promote the release of this carboxyl group as CO2. Then let go. And so now you get dopamine and you've regenerated your enzyme. So another thing, so pyridoxal phosphate is actually derived from vitamin B6. So remember I said that sometimes the cofactors were often derived from vitamins. And so when we talk about vitamins, typically like B6 is actually means like there's like a whole family of different compounds that are similar but not identical, that can all be considered B6, and some of them can be like interconverted between them and stuff, and it gets really confusing. But so basically this is a product of B6. And you also, this DOPA decarboxylase, so this DDC, it also decarboxylates other aromatic amino acids. So remember, aromatic means it has these rings. And because of this, another name for it is aromatic amino acid decarboxylase, or AADC. Okay, but the result is you get dopamine. And so some cells in their brain, so in a part called the substantia nigra, will stop there. They'll use dopamine as a neurotransmitter. And this is involved with like reward responses. But most of the cells will take things further. And the next step is going to be to do another hydroxylate, add another hydroxyl group. And so they're going to do it here. And this is going to be done with the help of dopamine beta hydroxylase. And so it's going to use ascorbic acid, which is vitamin C. And so it's going to hydroxylate here. And this is going to give you noradrenaline and norepinephrine. And so remember, this is going to be used extensively as a neurotransmitter, and some as a hormone. And most cells will stop here, but not in your adrenal gland, in your adrenal medulla, where they have the enzyme that they need in order to do the conversion. And so that enzyme that they need is this phenylethylamine N-methyltransferase, or PNMT. And it's going to use a cofactor too. And it's going to use a cofactor that I've talked about before too, S-adenosyl methionine. And so as the name suggests, this is coming from methionine. And this is coming, so it's coming from the amino acid methionine. So now we've talked about phenylalanine, tyrosine, lysine, and now we're on methionine. So methionine, it has the sulfur, and it has, you can see that it has this methyl group at the end after that sulfur. And it's kind of like begging to be taken off, isn't it? And so a methyltransferase is able to actually, methionine adenosyl transferase, or MAT, is able to take methionine, take ATP, and actually put this methionine onto this this adenosine part. So not the phosphates, but the phosphates will get released, and so this is going to provide energy to drive the reaction. And so this now is attached to this. And now you have a charge here, so you have something that's going to be more reactive. And a methyltransferase is going to help now transfer this carbon group to something else. And so now you can see that the sulfur, now you have lost the charge. So this is happier. And now your thing is methylated. And then the whole thing can be like regenerated. And so in this case, what we're methylating is we're methylating this amine group. And so this, now we have adrenaline. And this can be made by in your adrenal medulla and some of those, a few of those cells in your brainstem, and be really important for sending messages. And it's also important that you're able to degrade those messages. And so breaking down the catecholamines requires this, like catecholamine O-methyltransferase, or COMT, and monoamine oxidase, or MAO. And so you might have, MAO might sound familiar, and it's likely because MAO inhibitors can be used in some antidepressants. So tyrosine can be used to make a lot of different things, and other amino acids can be used to make other hormones. So like tryptophan can be used to make like serotonin. So there's all different uses of amino acids outside of just being for protein. So we've seen how we can have them being made to make hormones and to make like cofactors, like how methionine is used to make this SAM. And so yeah, so amino acids are even cooler than you thought.